Good day, ladies and gentlemen, and welcome to the President's Council Artemis Project conference call. I would now like to introduce your host for today's call, NBCC President Fran Visco. You may begin.
Thank you, and thank you to everyone who has called in, and thank you for your support of our work. I'm going to talk a bit about the Artemis Project and give you an overview, and then we're going to hear from two of the scientists who volunteer their time to work with us on this project, Greg Hannon from Cold Spring Harbor, and then Kim Lyerly from Duke. But let me talk a bit about what we're trying to do with our Artemis projects. That's sort of an umbrella name for the catalytic collaborative research work that we're doing within the Deadline campaign. We are identifying big questions in breast cancer that we believe are necessary to answer to achieve the deadline, and then bringing together individuals in the scientific community, advocates, policymakers, regulators, bringing them together as appropriate to design and implement plans of action to answer those questions. We want to focus on questions that are not being asked at a very high level elsewhere, but that we believe are very important. So when we started the Artemis Project, we made a decision that we would sort of pilot test the approach. And we wanted to ask the question, can you ask the question if there could be a preventive vaccine in breast cancer? Do we have the knowledge, the tools, and the technology to address that question? Not presupposing that we know what the answer is, but just wanting to know, does it make sense to look at that question at this time? We were very fortunate in that the National Philanthropic Trust agreed to fund 50% of the project, and so we were able to move forward. We had an initial meeting where we brought together a small group, about 15 individuals, to ask that initial question, does it make sense at this time? And the consensus was, yes, we can ask the question. Not a consensus that it will work, but yes, we have the tools to be able to ask the question. So following that, we developed an infrastructure that continues through which the process can occur. We identified and brought together a really great collaboration of very different types of scientists. We have some scientists who are ongoing participants and in a leadership capacity. There are advocates who are involved. We are in touch with the FDA and have had FDA representatives at our meeting. So we have the right people at the table to help address this issue. The process and the infrastructure that we developed includes developing a research plan and getting input on how that research plan should change from year to year, who should be at the table to do so. We have annual meetings, and during the annual meetings, and the number of people who participate in these meetings changes and grows each year, depending on what happened the prior year. During the annual meeting, we report out on what has happened. We identify issues that need to still be addressed, and we develop a plan of action for the coming year. There are reports of these meetings that exist on our website. For the issues that are identified, throughout the year we hold one or two small meetings. For example, last year we brought together a group to look at issues in immunology and in the immune system that had been raised at the prior annual meeting and that really needed a bit more focus. And so we bring smaller groups together around issues that have been identified at the annual meeting and develop plans of action to address those. In addition, we host one or two webinars a year where one of the participants will present their work to the larger collaboration and get input and answer questions. We also have a website where information and data are shared among the collaborators. And we, through the generosity of NPT, give small seed grants each year that will fund work that we believe is necessary to happen in order to push the work happening in Artemis to the next level. This is not a project that writes large grants to scientists to fund the vaccine development. The vision for this is that the seed grants will allow the scientists to get started and that the funding will come from other funding sources. We have given 2 seed grants to date looking at identification of targets, one to Paul Spellman and one to Paul Ewald. And what those seed grants will fund it is available on our website. In addition, there are advocate projects. As you know, we have Project LEAD, our science training project for breast cancer advocates. And Project LEAD graduates are at the table as collaborators in the Artemis Project, as they are in everything we do. And we have identified which projects come up at each meeting that advocates can take on. This year, for example, they are looking at other primary prevention trials to see what biomarkers were identified, what endpoints were utilized in those trials, and they are developing those data for the larger collaboration. That is just one of several projects that advocates are taking on. So in the Preventive Vaccine Artemis Project, we've identified 4 areas around which this collaboration has been formed. One is to identify the targets for a vaccine, and the 2 seed grants that have already been given out were in that area. The second is to look at the immune system, what the vaccine has to do. The third is to look at the issue of safety, because remember, this is a preventive vaccine, which means it is going to be given to a healthy population, so we have to be certain that it is safe. And we need a plan to make certain that happens. And then the fourth is efficacy. How is it that we will prove, and what do we want to prove to make certain this vaccine actually works? Today we're going to hear presentations on the first 2 issues, on the identity of a target and on the immune system, what the vaccine actually will have to do. It's important to understand that while this is happening under our leadership and we are supporting the infrastructure and the process to make this happen, and it's advocates who are really pushing this along and are collaborators in every sense of the word, we are working with an incredible group of scientists who are volunteering their time to be a part of this project. Truly high-level scientists who are giving their time to us, to the National Breast Cancer Coalition, to help answer this question. Because most of them believe in what it is we do. Many of them are intrigued by being given the opportunity to ask and answer a really difficult, unusual question. And every one of them enjoys working with a trained and educated advocacy community. So I want to turn now to 2 of the scientists who have agreed to talk to you today. And we're going to start with Greg Hannon from Cold Spring Harbor, who's going to give you a bit of background on what we're doing in target identification. Greg.
Hey, Fran. I guess the high-level scientists couldn't be here today, so you called me. Is that what happened?
That's what happened.
Okay, good. All right, presentation might be kind of a loose terminology for this, because what I'm really going to do is maybe just talk off the cuff a bit about the process that we've been using and the goals we've been trying to meet. And then hopefully, if people have questions, we can dig back into some of the details. And so, Fran, I guess I have about 10 minutes. So I'll start by saying that I got recruited to this project a few years ago, really as a skeptic. I came in. Fran had already convened a couple of meetings on this. The NBCC had held some discussions, and I came in really as someone, I have to say, who was probably biased against this being possible. And through sitting at these meetings over the years, and I guess the reason that I came as someone who was biased against this being possible was that it was a really intriguing idea. It's a grand vision, and I guess I've said this before, but whether I believed it or not, I felt that this is something that if it wasn't tried by the NBCC, it wouldn't be tried by anyone, probably ever. So I became increasingly convinced over the years that this is at least a possibly achievable goal, and I've even recruited some former lab members to come and also volunteer their time for this, because I do believe that it's something that if we bring the right people together, at least we're going to do the best possible job of evaluating the possibility that this could eventually happen. And so, obviously, people understand the generalities of vaccines, which is that you arm a person's own immune system to recognize and fight an invader. And in the case of infectious diseases, the successes are many and are obvious. Where you have something that is foreign to the body, you predict that if a person is exposed to this, then a disease will result. And in many cases, we've developed essentially successful vaccination strategies in those contexts by identifying components of the invading organism that the body can be taught to recognize and fight before an infection becomes established and becomes pathological. So the challenge with cancer, as opposed to infectious diseases, is much greater, because a cancer is essentially derived from a person's own tissues. And so the composition, the proteins, et cetera, that represent that disease are native to the person. And in other words, the person would not recognize those as being foreign because they are self proteins. And so the trick here is to identify components of the cancer that are either unusual, in other words, not presented normally to the immune system because they are the result of alterations that are cancer cell specific. Those alterations could be mutations, changes in the genomic sequence that are acquired by the cancer that cause it to produce a protein or a protein variant that is not normally present in a person's body. They could be changes in the amount of a particular component, an antigen, a protein that is expressed. In other words, if you have something that is present generally at a very low level, it is possible that a person could be trained to recognize that if it were presented at a much higher level than usual or in a different cell type or context than usual. There are certain proteins which might be expressed in ways that the immune system of a woman never sees them. This is also a set of possible antigens. In other words, there are things called cancer testis antigens that are normally present only in testis, but are reactivated because of aberrant gene regulation in cancers. That could be something that is a foreign, while not being foreign, yet still recognized as a foreign thing that the body could be trained to recognize. And we're still examining the possibility of an infectious agent etiology for some breast cancers. So really the challenge of the antigen identification group, the challenges have actually been many, some of which are imposed by the timeline under which the campaign is operating, the deadline 2020. And that does limit in some ways our ability to go and search for really long shot, let's say infectious disease causes of the disease of breast cancer, because we just don't think that within the time frame we could conceivably establish a sufficiently strong scientific case to nominate these agents as an antigen that is going to go in as a vaccine into healthy women, as Fran said. And so we've been sort of running through all of the possible ways in which we could identify candidate antigens. This has been happening sort of as a real sort of on-the-ground grassroots project over the last couple of years. It's been constituted into a group of volunteers probably over the last when was the meeting? Probably the last 4 or 5 months. The set of volunteers have been investing their own time and effort into mining public data on breast cancer, looking for candidate antigens, and that's leading to the development of some proposals, some of which will be funded, we hope, through the National Philanthropic Trust and through the organization and through the organizing efforts of the NBCC. But also, we hope that some of those proposals can be funded sort of more generally through peer-reviewed scientific funding to try to leverage the resources that the NBCC can bring to the table to really create, where the NBCC really creates the core, the seed of the program, and then it grows organically from there. There is a lot of publicly available data that one can mine to look for these kinds of potentially protective antigens. That data is being examined now, and there should be at least some initial idea of the level of promise that is present in the public data, probably in the next few months. I think our initial goal was somewhere around the end of summer to have some concrete answers derived from real-world data sets, but also during that time to develop the plans for any other types of data and analysis that we would need to gather in order to make nominations of candidate antigens that could then be evaluated by the sort of immunology groups and meet the kind of criteria that I suppose Kim is going to tell us about in a little while. We've got groups from Cold Spring Harbor, groups from Oregon Health Sciences University, groups from the Whitehead Institute at MIT, really all bending their expertise, again in a voluntary way, to this problem. It's resulted in a very unusual circumstance in science where a number of different labs come together on a collaborative and a voluntary basis. We have conference calls. We're shooting for about once a month to discuss our progress on this. It's leading to a lot of new method development for the kinds of data sets that are out there, partly being spearheaded by a former graduate student of mine who's now at the Whitehead Institute at MIT. And we're very hopeful that some candidates are starting to emerge. But just as important, and perhaps even more important, is the elimination of some of the possible routes toward a vaccine candidate. We started off this project, like every project, with a whole palette of possibilities. And as important as it is to rule things in, it is perhaps more important to rule things out so that we can narrow our focus and drill more and more deeply into candidates that are beginning to emerge. So I hope I've given you some idea of the sort of process that we're taking, both the analysis of data that's already been generated, but also the generation of new data and new data sets. And the challenge, of course, here is a very large one. We need to find something that we can train the body to recognize as an aberrant event to help it prevent breast cancer, but it has to be something that is not going to be deleterious, where that recognition is not going to be deleterious to a patient, a normal woman, over the course of her lifetime. And so we have a special responsibility here that is far beyond what one could imagine in a therapeutic setting. So, Fran, does that cover what you wanted me to cover?
Yes, it does. But I have one question that I'd like you to address.
Okay.
That is, when you talk about the publicly available data sets, could you explain what you mean by data?
Oh, okay. I'm sorry. I should have done. So basically what we're looking for here is large-scale measurements from tumors and from normal tissue types of really two sorts. One is expression levels of RNA which will code for proteins, and it's those proteins which will become the candidate antigens. And there we're looking for a protein being made in incorrect amounts or in an incorrect context, and that's sort of the theme of these cancer testis antigens that I mentioned. And then the second is we're looking at the quality of the RNA that's expressed, which will determine the content of the protein. And there we're looking for variations from normal, either acquired through mutations that characterize the tumor or acquired through alternative events that give rise to RNAs with a different content, a different sequence content than you find in normal cells, which would then generate different proteins which would appear non-native, even though they could be expressed without genetic changes in the program that directs the synthesis of those RNAs. Is that what you're?
Yes, that's it, exactly.
Okay, and there are large numbers, there are large amounts of data out there. They're not necessarily from ideal specimen types, but there's enough data out there that we can at least kick the tires on some of our ideas and approaches. And so I think all of us are suspicious that we will not be able to come fully to candidates from data sets that already exist. But we are quite confident that we can get fairly far along this sort of narrowing, this winnowing of possible approaches using data sets that are already generated. And that's good, because the generation of these data sets is a multi-tens of millions of dollar project.
Thank you very much, Greg. And Kim, your turn.
Well, thanks, Greg, and thanks, Fran, and again, welcome to everyone who's on the line. And again, I'd be happy to take questions after we've had our discussion. One of the things that I would like to start out by saying is that the process that Fran has described, that seems so clear and seems so logical, is really remarkable, because it's not really the strategy that most scientists are designed to implement. And what I mean by that is that many of the fundamental processes of training involve original ideas, original strategies to think about things, and innovation to do things that are quite different. And there's a lot of really important features of all of those aspects of science, but balancing this is the opportunity to converge scientists that have different skill sets, different ideas, different approaches, and have them, sort of develop a harmonized, mission-driven view. And one of the things that the NBCC has done is create a space, a domain that's highly reliable, authoritative, best-in-class, that allows these scientists that have a variety of skill sets and knowledge to interact with others that may not have the exact same skills, which, kind of our typical herd sort of mentality, and it allows us to extend ourselves and to freely interact in ways that are quite meaningful. So as Greg described, the use of genetics and the most up-to-date genetic strategies to identify differences in cancer cells, and there's obviously it leverages the billions of dollars spent by the U.S. government and other federal and international agencies to understand the cancer genome. What we're trying to do is leverage the insight and information and understanding how breast cancer is different and how it originated, to try to trigger what we call protective immunity. What is it about our bodies that does protect us from certain diseases? And perhaps 10 or 20 years ago, we couldn't have answered those questions, but today we have some very powerful indicators that we can answer those questions. So number one, cancer vaccines in general, preventative vaccines, clearly work, and there's evidence that hepatitis B, human papillomaviruses, these are external, these are foreign materials, if you will, but there are vaccines that have been developed that are clearly effective in reducing the rate of cancer. And so, that principle that you can develop a vaccine and protect against cancer, that is firmly established now, and again, probably more prominent over the last couple of years. Two, there's a recognition that just because there's a genetic difference, that doesn't necessarily mean that our immune system is going to fight those things off. And it turns out there's a lot of insight into the immune system and how it works and what its role is. For example, there are many times things are very different in our bodies, and our body doesn't reject them. For women who have babies, these babies, oddly enough, can be inside your body, but funny, you actually couldn't, in many cases, take a kidney transplant from your own baby because you're too foreign. Why could a baby develop but not be used in being recognized as foreign? So the molecular insights and the strategies in which the body turns immune systems on and off to appropriately recognize things that are foreign and should be fought off, and also turned off to not inappropriately, either through normal processes or through disease, get turned on. That's emerging. And in fact, just in the last couple of years, the checkpoints, if you will, the control mechanisms of the immune system have been identified, and new medications that turn on and turn off these control mechanisms are emerging. Now, what's interesting is that these control mechanisms turn on the general immune response against anything foreign, and they're noted to have very, very powerful anti-cancer effects. And clearly what that does is it helps us think about new cancer therapies and so forth, and that's terrific. But it also gives us the insight to say, well, gosh, rather than trying to generally turn on the entire immune system, why don't we turn on the immune system to specifically attack those elements of the body, those elements of the breast that are beginning to turn into cancer, and why don't we prevent that from happening? And that prevention, that strategy in which the body prevents tissues from changing, tissues from that contain, for example, mutations or abnormal expression of genes, if you will, too much of one thing, not enough of another. How can the immune system recognize that what is the most effective way to maintain and have an immune response that appropriately recognizes just those components that are foreign? That's really sort of where we are. So we'll create almost an incredible marriage of scientists that would generally not speak to each other, people who were working in genetics and proteomics, as well as people who are now immunologists, looking at the checkpoints and the cell biology of the immune system, working together to define and find preventative vaccines against the cancer. Now, as you can imagine, this approach is not for the faint of heart, and for many people, particularly those who control assets and want a return on investment, the timeline for some of these projects is much greater than the 3 to 5 years that people expect to return something on their investment. So much of the information that is being discovered will be applied to metastatic cancer or to late-stage cancer, because the hope would be you could turn around the disease, or you could show an impact on the life expectancy of someone who has 6 months or a year to live sooner than a preventative breast cancer vaccine. But the knowledge and the insight that is going to be gained in our work in the immune system, coupled with the new insights into what really is the initial events in breast cancer, what are the specific activities that are occurring and how can we prevent them, that sort of combined approach, the long-term view that's necessary to make a reality of the discoveries and the science into real products, that really is, I think, the umbrella of the NBCC and the Artemis Project, and we're very excited about the potential opportunities to continue the alliance and the collaborative efforts between not only the scientists but as Fran had mentioned, the regulatory bodies, the clinicians, and clearly the patient advocates that allow us to really recognize that this long-term goal will have an enormous long-term impact.
So thank you, Kim. So we had 2 questions that were presented by people who RSVP'd for the call, one of which I think Kim, you really responded to, and that is the therapeutic possibilities of the search for a preventive vaccine and a preventive vaccine. And the other question is one that we get asked often, and that is that we all know now that there are different subtypes of breast cancer, and the belief that they have substantially different biology. which I'm not sure about the substantially different biology point, but the question is, will a vaccine likely work for all types? And part of the question goes to the philosophy and vision of the Artemis Project and the National Breast Cancer Coalition. We started the Preventive Vaccine Project and maintain it with the concept that the goal is to find a vaccine for all healthy women to prevent breast cancer. Through the process, we may discover that there are vaccine approaches that are appropriate for a particular subtype of breast cancer over another subtype of breast cancer. If that is the case, we will certainly pursue each of those findings. But there is nothing that has come up that I know of, and Greg and Kim can correct me if this is wrong, but that says that it's impossible that there is an approach that would prevent all types of breast cancer. Is that correct, Kim and Greg? We don't have any proof that would be impossible.
Kim, do you want me to answer, or you?
Whoever. Go ahead.
Well, again, I would say there are already preventive vaccines. For example, the cervical cancer vaccine is designed to prevent, in one of the products, 4 different types of causes of cervical cancer that are caused by 4 different causative agents. So the idea that a formulation, if you will, or a component vaccine that includes multiple components, so you really, essentially, if you disaggregated that, you would have, essentially 4 different vaccines, but you can design them to serve as, single vaccine. So I think we can expand that. There's very likely that vaccine technology will allow us to, today, could immunize against 10 to 20 different cancer types. So, theoretically, there's no reason why we couldn't, include all of these different variants as the universal breast cancer vaccine. I guess getting to the point, and this is where Greg may want to weigh in as well, the idea is there some form of breast cancer where it would be, there's a process of cancer development that would make it theoretically impossible for an immune response to actually prevent it? I don't think there is. I don't think there's anything that I've seen where there's not a genetic change, either an amplification, a translocation, a mutation, and so forth. The only thing theoretically that could happen is that if you had to delete genes from cells, could that in fact create a cancer type cell from just loss of genes, and that would be an immunologically difficult thing. But it turns out that if you lose many components of the immune system, your body has a default mechanism to eliminate those cells anyway. But that would be the only, again, in theory, strategy where tumors could escape. But Greg may have some insight on the genetic side.
Well, again, I think this is a nuanced question, right? So I agree with you that there's not anything that theoretically says that there is not a way to create a vaccine against multiple, if not all, breast cancer types, though I doubt you will ever cover all breast cancers, all routes to developing breast cancer with a single vaccine. And it depends on a couple of things which have yet to be known. Certainly when we think of mutations, there is a skepticism emerging that recurrence rates are high enough to cover a whole broad spectrum of tumors, given bona fide genetic changes in the coding sequence of cancers. But regular changes in RNA content that are not driven by mutational events may be more prevalent and widespread. And so I think that there is still hope that we will cover a substantial swath of breast cancer subtypes with a number of antigens that could be fitted into a single vaccine. I mean, is that where you sit also on the conclusions that have been emerging, Kim?
Yeah, again, I think that's generally true. Again, we also, as we've talked, for those people who know about the different molecular subtypes, for example, there is a subtype of breast cancer called triple negative, And so some people might assume, oh gosh, if everything is negative, then how would you develop a cancer vaccine against something that doesn't have these antigens on them? Well, it turns out that the triple-negative breast cancers have plenty of mutations and plenty of antigens on them. They're just not the three that are actionable right now with anti-hormone therapy or anti-EGFR or B2-based therapies. So that would be the more simplistic view. And your view, your detailed sort of description, Greg, really I think covers, and I agree with your comments about the nuanced view of genetic change and coverage of a vaccine.
So let me ask a question that, may not make any sense, but it's in my head. So is there a reason to believe that whatever initiation step there is that ultimately leads to breast cancer, that initiation step, it's already determined at that point what type of breast cancer you're going to get? Or could it be that there is a fairly, similar initiation step for 95% of breast cancer, and what determines the type happens sometime after that?
You said your question wasn't going to make any sense.
Don't I always say that?
You do. I would say that we don't know yet, and this is part of the interest in looking at early lesions, right?
Right.
And so, in some ways, one could say that, there is a school of thought that would say that the initiating lesions drive the development of different breast cancer subtypes because they happen in different cell types within the mammary gland. And, that's a very sensible argument. And it fits with a lot of, I think, what we know about the molecular genetics of breast cancer. However, we see more and more that the different cell types are plastic and to some degree interconvertible. So it could be that early lesions happen in a much more common way than is currently suspected, and that something about the physiological course of disease determines the outcome. If you were to ask for my gut feeling, I would say that it's somewhere in the middle, that both of those extreme philosophies are incorrect. I have to say that I lean a bit more at the moment still toward the idea of some differences in cells of origin that, if not predetermine, at least predispose the development of the different subtypes. But I'm open-minded to the other possibilities. Kim, do you want to comment?
Well, I think Fran in her way can ask a very profound question. What are the origins truly of breast cancer, these different subtypes? I would say people have different views. For example, there's the perspective that breast cancer arises from a common cancer stem cell, and is it possible that since the stem cell can differentiate into a variety of different tissues, could in fact a cancerous stem cell develop into a variety of different cancer types, and would it be possible to generate a vaccine against the stem cells? Because of course that would be interesting but potentially more challenging. But I would say again, even in that case, if that ultimately turned out to be the biology of breast cancer, there's no theoretical reason why targeting antigens on cancer stem cells shouldn't be effective. I would say, again, in sort of support of what we're understanding about cancer, there's a real sense that there's an interaction between cancer cells, what are called the epithelial component of cancer, and the stromal cells, the fibroblasts and immune cells that are the microenvironment in which these epithelial cells grow. And it's possible that the genetic instability associated with cancer, which make it really challenging to develop therapies for and vaccines against, there may be constant elements within the stromal compartment that could be targeted by vaccines that could have an effect. So the idea that we've mostly focused on on the specific cancer cells and developing or the pre-cancer cells, developing a vaccine that prevents that from happening. But I, again, just wanted to mention that it's also possible that, and people are exploring this now, that the interaction between a cell and the specific interactions could be blocked, and that, in fact, may be another way to prevent cancer. You know, you're taking, you don't kill the engine, but you just take away the fuel line to the engine, and that's a similar success story.
Thank you.